Welcome back to the deep dive. We are shifting gears pretty significantly today. We're stepping out of the world of, textbook theory, those abstract lists of pathogens and diseases, and we are walking virtually through the double doors of the microbiology laboratory.
It really is a total shift in perspective.
Uh-huh.
I mean, until now, we've talked about bacteria as these sort of invisible antagonists in a story, but today we have to deal with the physical reality of them.
Right.
How do you actually find something that is completely invisible to the naked eye? And, once you do find it, how do you prove it's the killer and not just some innocent bystander that happened to be at the scene of the crime?
That is the absolute core tension of this deep dive, isn't it? We are covering what you could call the eyes and the soil of diagnostic medicine. We're talking about microscopy, literally how we see these things, how we translate that biological scale into a meaningful structure, and then in vitro culture.
Which is the art of persuading these microbes to multiply so we can actually get our hands on them and identify them.
Exactly. And for our structural spine today, we are walking through Murray's Medical Microbiology, the ninth edition, specifically Chapter 4.
And we'll be pulling in some really crucial clinical context from Javets, Melnick, and Edelberg as well, mostly from their chapters 5 and 47. Because, let's be honest, knowing how to grow a bug is, well, it's useless if you don't know how to interpret that result in the context of a real patient.
Let's start with that fundamental premise of the invisible world. It's so easy to take this for granted now, but for most of human history, medicine was just flying completely blind.
Oh, absolutely. I mean, you had concepts like miasma or bad air. The idea that a specific, discrete microscopic agent could cause a specific disease was a revolutionary leap.
And it was a leap that was entirely dependent on technology.
Entirely. You cannot conceptualize what you cannot see. The invention of microscope didn't just let us see small things. It fundamentally re-architected the entire process of medical reasoning. It turned medicine into a detective story where, for the first time, we had the ability to, dust for fingerprints.
So let's get into the hardware. The detective's magnifying glass. Murray starts, as he should, with the absolute workhorse of the lab, bright field microscopy.
Yeah, this is the standard. If you walk into any hospital lab anywhere in the world, this is the scope that's sitting on the bench. It's called bright field because, well, the name says it all. Light passes directly through the specimen, then through a series of lenses.
So the background is bright white, and the specimen, if it absorbs or blocks some of that light, appears darker.
That's the basic principle. Simple, effective. But, and this is a big but, there is a hard limit to this technology. You can't just keep adding stronger and stronger lenses and zoom in infinitely like you see in some sci-fi movie.
You eventually hit a wall.
You hit a wall made of physics. It's a concept called resolving power, or resolution. And resolution isn't just about magnification. It's not just about making things bigger.
It's about clarity.
It's about clarity. It's the ability to tell that two distinct points are actually two separate points, and not just one big blur. And that limit is fundamentally determined by the wavelength of visible light and something called the numerical aperture of the lens. For a standard bright field microscope, that limit is about 0.2 micrometers.
And that number, 0.2 micrometers, that is an absolutely critical threshold for you to memorize. It's one of those high-yield facts because it defines the boundary of what we can and cannot diagnose with a standard scope.
It really does. I mean, think about the scale. Most bacteria we deal with, Staphylococcus, E. coli, Salmonella, they're all roughly in the 1 to 2 micrometer range. So they are well above that 0.2 limit. We can see them pretty clearly.
But viruses?
Viruses are a different story. A typical virus might be, 0.02 to 0.1 micrometers?
Yeah.
They are orders of magnitude smaller. They're effectively invisible to bright field microscopy.
So if you're in an exam and you get a single best answer question about diagnosing a viral infection like influenza or measles, and one of the options is gram stain or bright field microscopy,
you can immediately, without hesitation, cross that out. It's the wrong tool for the job. You're trying to catch a minnow with a tuna net. It is just not going to work.
Okay, but even for bacteria, which we know are big enough to see, there's still a practical problem we have to overcome, right?
There is. And it has to do with how light behaves. As light passes from the glass, slide into the air, and then from the air into the glass of the objective lens, it bends, it refracts.
Because glass and air have different densities, different refractive indices.
Exactly. The refractive index of glass is around 1.5. For air, it's pretty much 1.0. That mismatch causes the light rays to scatter and disperse. Now, when you're at low magnification, say 10x or 40x, it doesn't matter all that much.
You don't really notice it.
You don't. But when you crank it up to 1,000x magnification, which is what we absolutely need to see bacteria clearly, that scattering becomes disastrous. You lose so much light, the image quality just falls apart.
And this is where the famous oil immersion technique comes in. I still remember doing this in my first micro lab, putting that little drop of sticky oil right on the cover slip and carefully lowering the 100x lens right into it.
It feels so counterintuitive, doesn't it, to deliberately smear oil all over a very expensive lens. But the physics behind it is, frankly, brilliant. Immersion oil has been engineered to have a refractive index of about 1.51.
Which is almost exactly the same as glass.
Precisely. So by bridging that tiny air gap between the slide and the lens with oil, you effectively eliminate the air interface. The light now travels through the slide, through the oil, and into the lens as if it were one continuous, unbroken column of glass.
It stops the scattering.
It stops the scattering and captures those peripheral light rays that would otherwise have been lost to refraction. This dramatically increases the numerical aperture of the lens, which, in turn, improves the resolution.
So what's the practical difference? What does that look like on the slide?
It's night and day. Without oil, a field of Staphylococcus looks like a blurry, indistinct purple smear. With oil, you can see the individual spherical cocci. You can see how they're arranged in those classic grape-like clusters. You can see the morphology. And morphology is always the first clue in the diagnostic puzzle.
Okay, so that's the workhorse. But sometimes bright field just isn't enough. Murray pivots here to dark field microscopy. This is a technique that, for some reason, always reminds me of driving in a really thick fog.
That's a fantastic analogy. I'm going to steal that. If you turn on your high beams in a fog, that's your bright field. The light just reflects straight back at you, and all you see is a wall of white glare.
You're blinded by your own light source.
Exactly. But if you have light coming in from the side,
You suddenly see the individual water droplets suspended in the air. Or another one I like is the dusty room analogy.
oh yeah, the sunbeam.
Right. You look at the air in a room, it looks perfectly clear. But if a single sharp beam of sunlight cuts across that room, suddenly you see thousands of tiny dust motes dancing in the beam.
That is the Tyndall effect, and that is precisely how dark field microscopy works. We use a special condenser with an opaque disc in it that blocks the direct central light from entering the lens. So the only light that the objective lens actually captures is the light that has hit the specimen and scattered at an oblique or sideways angle.
So the background remains pitch black and the organism itself glows, sort of like a star in the night sky.
It creates this incredible high contrast image. And the reason this is so critical for your exams and for real-world practice is that some bacteria are just too thin to be resolved by bright field, even with oil immersion.
They don't block enough direct light to cast a shadow, so to speak.
They don't, but they can scatter light beautifully.
And the classic suspect here, the one you absolutely have to know, is Treponema pallidum.
The agent of syphilis, yes. Treponema is a spirochete. It's shaped like a long, tight corkscrew. But it is incredibly narrow, less than 0.2 micrometers in diameter. So if you try to gram stain the fluid from a syphilitic chancre, you will almost certainly see nothing. The bacteria are there, but they're invisible.
But if you put that same fluid under a dark field scope,
Now you see them. You see these brilliant silver motile corkscrews spinning and flexing against that stark black background. It's unmistakable.
so high-yield clinical pearl. A patient presents with a painless genital ulcer, you suspect primary syphilis. The test of choice for immediate point-of-care diagnosis, if you have the equipment, is dark field microscopy of the lesion exudate.
That is correct, although I do need to add a small but important caveat that comes from Jowett's. You have to be extremely careful when using dark field for oral lesions.
Why is that?
Because the mouth is full of non-pathogenic commensal spirochetes that look an awful lot like Treponema pallidum. So dark field is fantastic for genital or skin lesions, but it's very risky for oral ones because of the high rate of false positives.
That's a great differentiator. Now, briefly, let's touch on phase contrast microscopy. This one feels a bit more niche.
It is, but it solves a specific problem. Phase contrast is for when you need to see the internal details of a microbe, but you don't want to kill it in the process. Most of our stains, the Gram stain, the Giemsa stain, they involve heat fixing or chemicals that kill the cells.
What if you need to see them alive?
Exactly. What if you want to study the life cycle of a fungus or watch the motility of a parasite in real time? You need them alive and kicking. Phase contrast cleverly exploits the fact that light travels at different speeds through materials of different densities.
So light moves slower through a dense nucleus than it does through the watery cytoplasm.
Right. And the microscope has special optical rings that convert those tiny differences in light speed or phase shifts into differences in brightness. It gives you this almost 3D-like high contrast image of a living, unstained cell. You can see the nucleus, vacuole granules all in real time.
Okay, so moving up the ladder of complexity, we get to fluorescent microscopy. If dark field is a sunbeam in a dusty room, fluorescence is like a neon sign in a nightclub.
That's perfect. It is the ultimate high-contrast technique. The core idea is that we stain the specimen with special dyes called fluorochromes. These are chemicals that have a unique property. They absorb light at one wavelength, usually invisible UV or blue light, and then they re-emit it at a different, longer visible wavelength.
So you shine blue light on them, and they glow green or yellow.
Precisely.
But why go to all that trouble? Why is this so much better than just using a regular colored stain?
It comes down to two things, sensitivity and speed. Let's create a scenario. Imagine you're a lab tech, and you're screening sputum samples for tuberculosis. With the standard acid-fast stain, the bacteria are tiny little red rods on a blue background.
A lot of a blue background.
A ton of blue background. You have to use the 100x oil lens. You have to meticulously scan hundreds of different fields of view to be confident that a slide is truly negative. It takes a long time, and eye fatigue is a huge problem.
It's like a microscopic game of Where's Waldo.
It is exactly that. But now, imagine doing it with fluorescence. The background is rendered completely black. The TB bacteria, stained with a fluorochrome like auramine rhodamine, glow like brilliant yellow-orange rods.
They just pop right out at you.
They jump out at you. And because the contrast is so high, you can use a lower magnification like 40x, which gives you a much wider field of view. You can scan the entire cover slip in a fraction of the time. If there's even one bright glowing rod on that slide, you will see it immediately.
So for screening high volumes of samples, which is common in public health labs, fluorescence is the undisputed efficiency king.
Absolutely. And it's not just for TB. We use it all the time for direct detection of viruses, too, by linking the fluorochromes to specific antibodies. But the core principle is always the same. Make your target glow like a firefly against a dark, empty void.
Which brings us to the final and most powerful tool in the arsenal, the heavyweight champion, electron microscopy. This is how we finally break that 0.2 micrometer barrier imposed by visible light.
We do. We completely leave the world of photons and enter the world of electrons. An electron beam, when accelerated, behaves like a wave, and its wavelength is roughly 0.005 nanometers.
Which is thousands of times shorter than the wavelength of visible light.
Thousands of times shorter. And since resolving power is directly related to wavelength, this means the resolving power of an electron microscope is thousands of times better. We can resolve structures right down to the molecular level.
This is how we actually visualize individual viral particles.
This is it. This is how we know that HIV is a sphere with spikes, or that Ebola looks like a shepherd's crook, or that a bacteriophage looks like a little lunar lander. We also use it to see the ultrafine internal structures of bacteria. Murray has some great images of this.
And there are two main types, right? Transmission and scanning.
Correct. Transmission electron microscopy, or TEM, looks through the specimen. The electron beam passes through an ultra-thin slice of the sample. It's for seeing internal structures, like ribosomes or the layers of a cell wall.
And scanning.
Scanning electron microscopy, or S, looks at the surface of the specimen. The beam scans across the surface, and we detect the electrons that bounce off. This gives you these incredible, detailed, 3D topographical images.
But let's be clear, we don't use this for routine diagnosis, do we? A patient comes into the ER with the sniffles, we don't wheel out the multi-million-dollar electron microscope.
Oh, heavens no. It's almost exclusively a research tool, or maybe for a once-in-a-career baffling outbreak investigation. It's incredibly expensive, it's technically difficult, and the sample preparation is brutal and time-consuming. You need to know it exists because it defines our fundamental structural understanding of the microbial world, but you won't be ordering it in the clinic.
Okay. So we've covered the lenses, the hardware for seeing, but before we can look at anything, we have to prepare the sample. You can't just, throw a chunk of tissue on a stage and expect to see anything useful. Let's talk about examination methods.
The simplest of all methods, and the starting point, is the wet mount. And it is literally what it sounds like. You take a liquid sample, say stool or vaginal discharge or urine. You put a single drop on a slide, maybe add a drop of saline to dilute it, pop a cover slip on top, and you look.
What are we hoping to see in this kind of raw, unprocessed state?
We're looking for things that are relatively large and have a distinct shape or, crucially, movement. In a stool sample, we're looking for the eggs or cysts of parasites, like Giardia, with its falling leaf motility, or the cysts of Entamoeba. In a vaginal swab, we might see clue cells.
which are epithelial cells just covered in bacteria, indicating bacterial vaginosis.
Exactly. Or you might see the characteristic swimming, spinning protozoan Trichomonas vaginalis.
And movement is the key here. Because we haven't killed them with stains or fixatives, we're seeing them in their living state.
That's the real advantage. And Javis makes a point to mention the hanging drop preparation here, which is a variation of the wet mount, specifically designed to observe bacterial motility. If you suspect an infection with Listeria, which has that characteristic tumbling motility, or Vibrio with its darting motility, seeing that in a wet mount can be very strong diagnostic evidence.
Okay, now, what if the sample itself is messy? Let's say you have a skin scraping from a patient with a scaly red rash. You suspect a fungal infection. But skin scrapings are full of, well, skin, keratin, cellular debris. Under a microscope, it just looks like a pile of rocks.
It does. It's completely uninterpretable. And this is where we use what I like to call the drain cleaner method, the 10% potassium hydroxide, or KOH prep.
Why do you call it drain cleaner?
Because that's essentially what it is. Potassium hydroxide is a strong alkali. You mix a drop of 10% KOH with the skin scraping on the slide. The alkali goes to work, and it attacks and dissolves the proteins. It digests the keratinocytes, the human cell membranes, all the background debris. It literally just melts away the human tissue.
Yeah. But it doesn't digest the fungus that we're looking for.
It doesn't. And this is the crucial biological differentiator. Fungal cell walls are made of chitin. Chitin is this incredibly tough, fibrous polysaccharide. It's the same stuff that makes up insect exoskeletons, and it is highly resistant to alkali. So after the KOH has done its work, all the human mess is dissolved, and you're left with these beautiful, clear, branching fungal hyphae.
So KOH prep is for fungi. Got it. Next up in Murray's list is India ink.
Right. This is a very specialized stain for a very specific clinical scenario. Suspected cryptococcal meningitis. Cryptococcus neoformans is a yeast, but it has a superpower. It's surrounded by this massive, thick, gelatinous polysaccharide capsule.
And India ink is what we call a negative stain.
That's the key. It's not a true stain that binds to the cell. It's actually a suspension of fine carbon particles. When you mix a drop of this ink with the patient's cerebrospinal fluid on a slide, the ink darkens the entire background. It makes it black. But the carbon particles are too large to penetrate that thick, slimy capsule of the yeast.
So the yeast cell creates its own little force field against the ink.
Effectively, yes. What you see under the microscope is a dramatic, starry night background. And against that black, you see these perfectly round, clear halos. And sitting right in the middle of each halo is the small, budding yeast cell. It's one of the most classic images in microbiology. If you see the word halo in a question about meningitis, the answer is almost certainly Cryptococcus.
Now, Murray does add a modern caveat here that antigen testing is actually more sensitive now.
He does, and he's right. Tests like the cryptococcal antigen, CRAG test, are more sensitive and specific. But the India ink prep remains a classic, rapid test and a perennial favorite on board exams. You still have to know it.
Absolutely. Okay, let's move to the main event, the most famous, most important procedure in all of microbiology, the Gram stain. This is the great passport system of the bacterial world.
It really is. Hans Christian Gram developed this technique way back in 1884. And it is arguably still the single most important and most frequently performed test in the diagnostic lab. It fundamentally splits the entire bacterial world into two great empires, the gram-positives and the gram-negatives.
And this isn't just some arbitrary color code. It reflects a deep, fundamental difference in their cellular architecture. We really need to unpack the mechanism here because so many students just memorize the colors, purple positive, pink negative, but they forget the why, and the why is what makes it stick.
The why is all about the peptidoglycan. This is the cross-linked polymer mesh that makes up the structural backbone of the bacterial cell wall.
So let's use a mental model. Imagine gram-positive bacteria as having a cell wall that's like a thick, dense, multi-layered sponge.
A very thick sponge, maybe 40 to 80 layers of peptidoglycan deep. It's a massive, robust structure. Now compare that to gram-negative bacteria. They have a very, very thin sponge, maybe just 1 or 2 layers thick. But, and this is the key, they have an outer raincoat, an extra outer membrane made of lipids and lipopolysaccharide.
Okay, so thick sponge versus thin sponge with a raincoat. Got it. Let's walk through the procedure. Step 1, crystal violet.
You flood the slide with this intense purple dye. It's water-soluble. It gets everywhere. It soaks right into the thick sponge of the gram-positives. It soaks into the thin sponge of the gram-negatives. At this point, everything is purple.
Step 2, grams iodine.
This is the mordant. A mordant is a substance that helps to fix a dye. The iodine enters the cells and binds to the crystal violet molecules. They form these large, insoluble crystals, these large molecular complexes that get physically trapped inside the mesh of the peptidoglycan sponge.
So everything is still purple, but now the purple's kind of locked in
It's locked in.
for now.
Then comes step 3, the decolorizer. This is usually alcohol or an acetone alcohol mixture. This is the single most critical step in the entire procedure.
This is where the difference in architecture really matters.
This is where it all happens. When the alcohol hits the gram-positive thick sponge, it has a dehydrating effect. It sucks the water out of the peptidoglycan, causing the entire mesh to shrink and tighten. Those large purple iodine complexes that were formed are now physically trapped. The pores of the sponge have closed down on them. They cannot get out. So the cell stays a deep, dark purple.
But what happens with the gram-negative, with its raincoat and thin sponge?
Well, what does alcohol do to lipids?
It dissolves them.
It dissolves them. So the alcohol hits that outer membrane raincoat and just strips it away almost immediately. Then the alcohol hits the very thin single-layer sponge underneath. It's way too thin and porous to trap anything. The purple dye that was in there just washes right out in seconds. The cell is now colorless and invisible again.
So if you stop the procedure right here, you'd have your purple gram-positives, but your gram-negatives would be completely invisible, which is why we need the final step. Step 4, the counterstain.
Right. We use safranin, which is a pink or red color dye. You apply that to the slide. The gram-positive cells are already stuffed full of that dark purple dye, so they don't really pick up the much lighter pink. They stay purple. But the now colorless gram-negative cells happily soak up the pink safranin and turn pink.
And there you have it. Purple is positive, pink is negative.
And this isn't just academic. For a clinician at the bedside, this is the first branch point in deciding empiric antibiotic therapy. A patient with meningitis. Is it purple cucky in pairs, suggesting strep pneumo? Or is it pink cuccobacilli, suggesting Haemophilus? The treatment is completely different. This one stain guides billion-dollar decisions every single day.
But of course, there's always a group of bacteria that refuses to play by the rules. The rebels, in this case, the mycobacteria, the agents of TB and leprosy.
Ah, yes. The waxy ones. They have a cell wall that is just loaded with these long-chain fatty acids called mycolic acids.
I always think of it like trying to stain a candle. If you pour a watery purple dye onto a wax candle, it just beads up and rolls right off.
That is a perfect analogy. The aqueous dyes of the gram stain simply can't penetrate that hydrophobic waxy layer. So mycobacteria just don't stain, or they stain very poorly and unreliably, sometimes called gram ghosts.
So we need a different approach. We need the acid-fast stain. We have to somehow force the dye into that waxy cell.
We do. And in the classic Seale-Nielsen method, the hot method, we do this with a combination of a potent dye and physical force. We use a red dye called carbofucin, which is lipid-soluble, and we apply heat. We literally steam the slide over a flame or on a hot plate.
The heat melts the wax.
It doesn't melt it completely, but it softens it. It increases the fluidity of that mycolic acid layer enough to allow the red carbofucin dye to seep inside.
Yeah.
Then once the slide cools down, the wax sort of rehardens, trapping the red dye inside.
And then comes a decolorizer, which in this case is a really brutal 1-acid alcohol.
Much, much stronger than the alcohol used in gram staining. This harsh decolorizer strips the red dye out of everything else on the slide. Human cells, other bacteria, debris. But the mycobacteria, with their waxy wall now holding the dye captive, hold on to it fast. They resist the acid, hence the name acid fast.
And then you counterstain with methylene blue.
Right, so everything else on the slide turns blue, and the acid-fast bacilli stand out as bright red rods against that blue background.
Now, Murray also mentions a cold method, the Kinyon method.
Yes, the Kinyon method is a modification that avoids the need for heating. Instead of heat, it uses a much higher concentration of phenol in the carbofucin dye. Phenol acts as a chemical detergent, helping to solubilize the mycolic acid layer and force the dye in without heat. It's much safer for lab personnel because you aren't creating aerosolized infectious TB bacteria by steaming them.
And we already touched on the fluorescent version, auramine rhodamine, as the go-to for screening.
Correct. That's the modern screening tool. The fluorochrome dye binds specifically to the mycolic acids. It's faster and more sensitive because you can scan at low power. But if you find a positive with fluorescence, you typically confirm it with a classic Seale-Nielsen or Kinyon stain to be absolutely sure.
Let's just quickly run through a few of the other specialized stains that Murray lists, the passports for the more unusual suspects.
Sure. If you're looking for blood parasites, especially malaria, you're going to use the right Geomesis stain. This is the standard stain used in hematology labs. It stains the cytoplasm of cells blue and the nucleus red or purple. When you look at red blood cells, you're looking for the classic signet ring shape of the Plasmodium trophozoite inside the cell.
What about for intestinal parasites from a stool sample?
For those, particularly protozoa like Entamoeba histolytica or Giardia lamblia, the trichrome stain is often used. It provides excellent visualization of the internal structures of the parasite cysts and trophozoites, like the number of nuclei, which is critical for species identification.
And a very important modern update that has largely replaced the simple KOH prep for fungi.
That would be chalcofluor white. This is a fluorescent dye that binds with high affinity to cellulose and chitin in the cell walls of fungi. It fluoresces a brilliant blue-white under UV light. It's incredibly sensitive and is now the standard for detecting fungi in tissue, and especially for Pneumocystis jirovecii, which is a major cause of pneumonia in immunocompromised patients, particularly those with AIDS.
Okay, we've covered a lot. We have looked at them, we have stained them, we have a primary suspect, but seeing a gram-negative rod on a slide doesn't tell you if it's a relatively harmless E. coli from the gut, or if it's Salmonella, a definite pathogen, or Pseudomonas, which can be a nightmare to treat.
Exactly. To know who the suspect is, you need to interrogate them. You need to see what they do, what they eat, how they behave. You need to grow them.
And this brings us to the second major part of the chapter, in vitro culture. We're moving from simple detection to identification and characterization.
And this is where the whole concept of the menu comes into play. You mentioned the term fastidious organism earlier, and I think it's a perfect way to frame this.
It's a great term. It just makes them sound so high maintenance.
They are. Fastidious simply means they're picky eaters. Some bacteria are incredibly rugged survivors. E. coli or Staph aureus will grow on almost anything. You could probably grow them on a piece of damp cardboard. But other bacteria are absolute divas. They have very complex nutritional requirements. If you don't give them exactly what they want, specific amino acids, certain vitamins, precise atmospheric conditions, they will simply refuse to grow. They'll die.
So the microbiology lab has to be a kind of gourmet kitchen with a whole pantry of different ingredients to satisfy these picky eaters.
That's a great way to put it. And we can break down this menu of culture media into three broad categories. Category 1. The buffet. These are the enriched, non-selective media.
And the absolute classic here is blood agar.
Blood agar, or BA, is the baseline for most cultures. It's a rich nutrient agar base that's been supplemented with 5% sterile sheep blood.
Why specifically sheep blood?
Good question. It's rich in nutrients, so almost all medically important bacteria will happily grow on it. It doesn't contain certain inhibitors that might be present in human blood. But just as importantly, the blood itself acts as a built-in diagnostic test. We look for hemolysis, the ability of the bacteria to produce enzymes that break down those red blood cells.
This is a absolutely crucial differentiator, especially for the different species of Streptococci.
It is the first thing you look for. And there are three main patterns you need to know, Cole. First is beta hemolysis. This is total, complete destruction. The bacteria produce powerful enzymes called hemolysins that completely rupture the red blood cells in the surrounding agar. What you see is a clear, transparent halo around the bacterial colony. You can literally read a newspaper through it.
And the classic beta hemolytics.
Streptococcus pyogenes, which is group A strep, the cause of strep throat, and also Staphylococcus aureus. Both are potent beta hemolytics.
Okay, then there's alpha hemolysis.
This is partial, incomplete destruction. The bacteria produce enzymes that damage the red blood cells and oxidize the hemoglobin, turning it into methemoglobin. This creates a greenish, bruised-looking halo around the colony. The classic alpha hemolytic organism you must know is Streptococcus pneumonia, a major cause of pneumonia and meningitis.
And finally, gamma hemolysis.
Which is a bit of a misnomer because it actually means no hemolysis. The bacteria grow just fine on the blood agar, but they don't produce any hemolysins. They don't touch the red blood cells at all. So the agar right up to the edge of the colony stays opaque and red. Many species of Enterococcus are gamma hemolytic.
So the food itself is a diagnostic test. Brilliant. Now let's talk about chocolate agar, which, to the disappointment of every first-year medical student, is not actually made of chocolate.
Not even a little bit. It's simply blood agar that has been cooked.
Yeah.
You prepare the blood agar base, and then you heat it to about 80 degrees Celsius until the red blood cells they pop open. This process turns the medium a muddy brown color, which I guess someone thought looked like chocolate.
Why would you cook the blood? What's the point of lysing the cells?
It's to release the hidden goodies inside. Locked within the red blood cells are essential growth factors, specifically NAD, which is also called factor V, and hemin, which is factor X.
And who are the picky eaters that need this pre-chewed, pre-digested food?
The really fragile, fastidious pathogens. Specifically, you have to remember Haemophilus, influenza, and Neisseria. These bacteria are, in a way, toothless. They are unable to produce their own hemolysins to break open the red blood cells themselves. So if you plate them on regular blood agar, they will starve to death, surrounded by food they can't access. Chocolate agar serves them those essential factors V and X on a silver platter.
So another critical clinical rule, if you suspect bacterial meningitis, which is often caused by Haemophilus or Neisseria meningitis, you must plate the cerebrospinal fluid on chocolate agar. If you only use blood agar, you could get a false negative and miss the diagnosis entirely.
100% correct.
Right.
It is non-negotiable.
Okay, that was the buffet. Let's move to category 2. Selective and differential media. I like to call these the smart filters.
A very apt description. This is where we start to actively weed out the suspects and force the ones that survive to tell us something about themselves.
Let's define our terms here. Selective versus differential.
Right. Selective means the agar contains some kind of inhibitor, a chemical bouncer, if you will, that kills or prevents the growth of the bacteria we're not interested in. Differential means the agar contains a built-in signal, a name tag, which is usually a color change, that tells us something about the unique biochemistry of the organisms that manage to survive and grow.
And the most high-yield, must-know example of a medium that is both selective and differential is McConkey agar.
Oh yeah, you cannot pass a microbiology exam without understanding McConkey agar. It is specifically designed to isolate and differentiate the gram-negative rods, particularly the enteric or gut bacteria.
So let's start with the selection part. What are the bouncers in McConkey?
The bouncers are bile salts and the dye crystal violet. Now think about where these bacteria live. E. coli lives in your intestine. It is bathed in bile every single day of its life. It is evolutionarily adapted to survive in that harsh environment. But what about skin bacteria like staph or strep? They never encounter bile. So if you put bile salts into the agar, the staph and strep and other gram-positives just die. Only the hardy gram-negative gut bugs can survive.
So if something grows on McConkey, you know right away it's a gram-negative rod. That's a huge piece of information. Now, for the differentiation, the name tag.
The name tag is the sugar lactose. This is a great biochemical divide in enteric bacteriology. The agar contains a generous amount of lactose and a pH indicator dye called neutral red.
And the logic is?
The logic is simple. If the bacteria can ferment lactose, it produces acidic byproducts. The acid lowers the pH around the colony, and the pH indicator turns a bright pink or red color. The classic lactose fermenters are organisms like E. coli, Klebsiella, and Enterobacter. They grow as vibrant pink colonies.
And the ones that can't ferment lactose?
They have to eat something else, so they metabolize the proteins, the peptones, in the agar instead. This process generates ammonia, which is alkaline. This raises the pH so the colonies remain colorless or translucent. And the kicker is that many of the most dangerous gut pathogens, Salmonella, Shigella, Pseudomonas, are non-lactose fermenters.
So when a tech looks at a stool culture on a McConkey plate, they're thinking, pink colonies are generally normal flora, with some important exceptions, of course, but the colorless colonies, those are the ones to investigate. That is a massive diagnostic fork in the road.
It is the first and most important observation they will make on that plate.
Let's look at another classic, mannitol salt agar, or MSA.
Okay, so if McConkey is built for gram-negative rods, MSA is built for Staphylococcus. The selector here is a salt, a very high concentration of it, 7.5% sodium chloride. Most bacteria, if you put them in that much salt, will shrivel up and die due to osmotic stress. But Staphylococcus species love salt. They live on our salty, sweaty skin. It's their natural habitat, so they survive and thrive.
And the differentiator in this case?
The sugar is mannitol, and the pH indicator is phenol red. The key differentiator is between the two major staph species. Staphylococcus aureus, the golden staph, the pathogenic one, is a vigorous fermenter of mannitol. It produces acid, which turns the phenol red indicator from pink to a bright yellow.
And the other big one, Staphylococcus epidermidis.
S. epidermidis, the much more common and usually harmless skin commensal, grows just fine on the high salt, but it does not ferment mannitol, so its colonies remain pink. The rule is simple. On MSA, yellow means aureus. Pink means epidermidis, or another coagulous negative staph.
Another really clever trap is XLD agar, which stands for xyloslicine deoxycholate. This one is specifically for stool pathogens.
This one is a bit more complex. It's like a little biochemical timer. It's selective because it has deoxycholate, which inhibits gram-positives. But the differential part is multi-step. First, it gives the bacteria a simple sugar to eat, xylose. Most gut bugs, including Salmonella and Shigella, can ferment xylose, and they initially produce acid and turn the colonies yellow.
Okay, so they start out yellow.
They do. But once they've used up all the xylose, Salmonella and Shigella have a special trick. They can switch to eating the amino acid lysine, which is also in the agar. They have an enzyme called lysine decarboxylase.
And that reaction changes the pH again.
Right back in the other direction, the decarboxylation of lysine produces alkaline byproducts that overcome the initial acid from xylose fermentation, and the colony turns back to red. So red colonies on XLD are suspicious.
But there's one more trick to differentiate Salmonella from Shigella.
The final clue. The agar contains sodium thiosulfate and ferric ammonium citrate. Salmonella species can produce hydrogen sulfide gas, H2S, from the thiosulfate. This colorless gas reacts with the iron in the agar to form a black, precipitate ferric sulfide.
So let's put it all together. The final pattern for Salmonella is a red colony with a distinct black center. Shigella is a red colony with no black center. It's an elegant, purely visual code.
And it allows a lab technician to look at a plate that might have 1,000 colonies of normal flora on it and instantly pick out the 2 or 3 colonies that are the likely pathogens. It's incredibly powerful.
Okay, let's move to our third and final category, the VIP rooms. These are the highly specialized media designed for the most high-maintenance, fastidious suspects.
Right. Let's just run through the absolute must-knows for exams. First, Lowenstein-Jensen medium, or LJ.
This is for TB.
This is for Mycobacterium tuberculosis. It's a very rich, egg-based medium. It's literally made with whole eggs and potato flour. It also contains malachite green dye. The reason for this is that TB grows incredibly slowly. It can take 4 to 6 weeks to see colonies. The malachite green is an inhibitor that prevents the fast-growing bacteria and fungi from the patient's sample from completely overgrowing the plate before the slow-growing TB even gets a chance to start.
Next up, Thayer-Martin agar.
This is the medium for isolating the gonococcus, Neisseria gonorrhea. Now think about the sample site for gonorrhea, a urethral or cervical swab. That site is absolutely teeming with normal Floralactobacilli staph yeast.
It's a very crowded neighborhood.
A very crowded neighborhood.
Yeah.
Thayer-Martin is essentially chocolate agar to provide the rich nutrients that the fastidious Neisseria needs, plus a powerful cocktail of antibiotics to kill all the competition. It's a selective medium on steroids.
What's in the cocktail?
The classic formulation has vancomycin, which kills gram-positives, colistin, which kills most other gram-negatives, and nystatin, an antifungal to kill yeast. It creates this perfect safe space where only the naturally resistant Neisseria can survive and grow.
Very clever. What about BCYE, buffered charcoal yeast extract?
The name gives you some clues. The target here is Legionella pneumophila, the cause of Legionnaire's disease. Legionella is another diva. It has an absolute requirement for 2 specific nutrients, iron and the amino acid L-cysteine. BCYE provides those. The yeast extract is a rich source of nutrients, and the charcoal is there to act like a detoxifier. It absorbs toxic metabolic waste products that would otherwise build up and kill the very finicky Legionella.
And finally, a very colorful one, TCBS agar.
TCBS stands for thiosulfate citrate bile salts sucrose. This is for isolating Vibrio species, most famously Vibrio cholerae. It's highly selective because it has a very high pH, is quite alkaline, which Vibrio loves, but most other bacteria can't tolerate. And it's differential based on sucrose fermentation. Vibrio cholerae ferments sucrose and produces large, bright yellow colonies.
That covers the agar plates. But as we know, some pathogens simply refuse to grow on any kind of dead artificial media. They require a living host. Which brings us to Section 6, cell culture.
We are now talking about the obligate intracellular pathogens. This includes all viruses, without exception, and some specialized bacteria like chlamydia and rickettsia. These organisms are metabolically incomplete. They lack the basic machinery to replicate on their own. They absolutely have to hijack the machinery of a living host cell.
So the medium in this case isn't agar in a petri dish. It's a living monolayer of human or animal cells growing in a test tube or a flask.
That's right. In the virology lab, we use established cell lines. These are often immortalized cells derived from tumors, like HeLa cells from cervical cancer or A549 cells from lung cancer. We grow them in a flask until they form a nice confluent sheet. Then we infect them with a patient sample.
And then we just watch. What are we looking for?
We watch them. We look for the CPE, the cytopathic effect.
A damage report.
Exactly. It's the visible damage that the virus inflicts on the cells. Does the virus make the cells round up, die, and detach from the flask? That's what you see with poliovirus. Does it make the cells fuse together into giant multinucleated blobs called syncytia? That's characteristic of viruses like respiratory syncytial virus, RSV, or herpes simplex virus. The specific pattern of destruction is a major clue to the virus's identity.
But waiting for a virus to slowly kill a sheet of cells can take days or even weeks. Javits mentions a clever technique to speed this up: the shell vial culture.
Yes, this was a huge innovation for clinical virology. It's ingenious. Instead of just adding the patient's sample to the top of the cells and waiting passively for the virus particles to drift down and attach, we use centrifugal force. We put the sample onto a cover slip with cells at the bottom of a small vial, and we spin it in a centrifuge.
You physically slam the virus particles onto the surface of the cells?
You do. This forces the infection to happen much more rapidly and efficiently. We can then let the virus replicate for just 24 to 48 hours and then stain the cells with specific fluorescently tagged monoclonal antibodies to detect the viral proteins. It turned a week-long test into an overnight result. It's the classic method for the rapid detection of cytomegalovirus, or CMV.
Speed is life in medicine. And that brings us perfectly to our final and maybe the most important section of this chapter, Section 7, clinical interpretation.
This is really where the science of the lab meets the art of medicine. You have a result on a piece of paper: growth of Staphylococcus aureus. But is that the answer to the patient's problem? Javits chapter 47 is dedicated entirely to this question. The core principle is diagnosis is a dialogue between the clinician and the laboratory.
Let's start with the most basic rule: garbage in, garbage out. The importance of specimen quality.
Oh, this is everything. The classic example that every medical student needs to understand is the sputum sample. A patient has pneumonia. You need a sample of the infected pus from deep down in their lungs. You ask them to provide a good, deep cough. They hand you a cup.
And the lab's first job is to vet that sample.
Their first job is to perform a quality check, a Gram stain screen. If they look under the microscope and see a lot of squamous epithelial cells.
Those are the flat, paving stone cells that line the inside of your cheek and tongue.
Correct. If you see those, it means the patient didn't cough up sputum, they just spit in the cup. That sample is saliva. It is clinically useless. It's full of normal mouth flora. If you culture that, you will grow a forest of irrelevant bacteria, and the patient might get treated for the wrong thing.
So what does a good sample look like?
The lab must reject that saliva sample. A good sputum sample has many polymorphonuclear leukocytes, PMNs, or neutrophils, and few or no squamous cells. The presence of neutrophils tells you the sample came from the site of inflammation deep in the lungs.
Okay, what about blood cultures? This is a huge source of anxiety and confusion for junior doctors. The dreaded phone call in the middle of the night.
Contamination is the #1 enemy of blood cultures. Your skin is covered in bacteria, most commonly Staphylococcus epidermidis. When you stick a needle through the skin to draw blood from a vein, you might drag a tiny plug of skin with it into the collection bottle.
So the lab calls you and says, the blood culture is positive for Staph epidermidis. You're faced with a critical dilemma. Is this true bacteremia a bloodstream infection, or is it just a dirty needle stick?
And how do you decide?
It's all about the pattern.
It's all about the pattern. This is why we always draw multiple sets of blood cultures, ideally from different venipuncture sites, over a period of time. If you draw 2 sets, meaning 4 bottles total, and only one of those 4 bottles grows Staph epi, it is almost certainly a contaminant. It's statistically very unlikely that a real bloodstream infection would only show up in 25% of the blood you drew.
But if 2, 3, or all 4 bottles grow the same bug?
Now that's real. That's a pathogen. That is true bacteremia, and you need to act on it immediately.
And finally, let's talk about urine. The numbers game.
Right. Urine inside the bladder is sterile. But by the time it leaves the body, it has passed through the distal urethra, which is colonized with bacteria. So a clean catch midstream urine sample will almost always have some bugs in it. The question is, how many?
We use a quantitative threshold.
We do. We take a calibrated loop of urine, plate it, and count the colonies. The magic number, the general threshold, is 10 to the 5th or 100,000 colony-forming units, CFU, per milliliter. If the count is above 10 to the 5th, we generally call that a true infection, like cystitis or pyelonephritis. If it's below 10 to the 4th, it's usually considered contamination from the surrounding skin.
But context matters here too.
Always. If you get urine directly from the bladder with a needle, a suprapubic aspirate, then any growth is significant. If the patient is a symptomatic male or has a catheter, lower counts, even 10 to the third, can be clinically significant. But for a routine sample from a female patient, 10 to the fifth is the number to remember.
We have covered an absolutely immense amount of ground today, from the physics of light bending through a drop of oil to the intricate biochemistry of sugar fermentation on a Petri dish.
It's a lot, I know. But if you remember the narrative arc, it helps. We make the invisible visible, that's microscopy. Then we feed the suspect and give it tests to make it reveal its true identity. That's culture. And finally, we interpret that identity in the context of the patient and the quality of the sample. That's clinical reasoning.
Fantastic summary. Okay, before we sign off, let's try to lock this all in. I'm going to pose 5 single best answer or SBA-style questions. I'll give you the scenario, pause for a moment for you to think, and then our expert will break down the correct answer and, more importantly, the why.
Let's do it.
Question 1. A laboratory technician is screening sputum smears for Mycobacterium tuberculosis. To maximize efficiency and screen the largest possible surface area in the shortest amount of time, which staining technique should be prioritized for the initial screen? A. Sil-Nielsen stain. B. Kinyan stain. C. Auramine-rhodamine stain. D. Gram stain. E. Hematoxylin and eosin stain.
The correct answer is C, Auramine-rhodamine stain.
And why is that?
Okay, so the key words here are maximize efficiency and screen the largest surface area. Auramine-rhodamine is a fluorescent stain. Because the acid-fast bacteria glow as bright yellow or orange rods against a perfectly dark background, the technician can use a lower power objective like 40x to scan the slide quickly. This allows them to see a much wider field of view compared to the 100x oil immersion objective that is required for the traditional bright field stains like Sil-Nielsen A or Kinyan B. The Gram stain D is useless for mycobacteria because of their waxy cell wall.
Perfect. Question 2. A stool sample from a patient with acute bloody diarrhea is plated on McConkey agar. After 24 hours of incubation, the plate shows abundant growth of colorless colonies. The presence of these colorless colonies effectively rules OUT which of the following organisms as the cause. A. Salmonella enterica. B. Shigella dysenteria. C. Pseudomonas aeruginosa. D. Escherichia coli. Proteus mirabilis.
The answer is D, Escherichia coli.
The
McConkey agar is a differential medium based on the ability to ferment lactose. Lactose fermenters produce acid, which turns the colonies pink or red. E. coli is a classic, strong lactose fermenter. Therefore, if the colonies are colorless, it means the organism is a non-lactose fermenter, and we can effectively rule OUT E. coli. All the other options, Salmonella, Shigella, Pseudomonas, and Proteus, are classic non-lactose fermenters and would be expected to produce colorless colonies.
Excellent. Question 3. A 19-year-old college student presents to the emergency department with fever, headache, and a stiff neck. A lumbar puncture is performed to ensure the isolation of fastidious organisms commonly associated with bacterial meningitis, such as Neisseria meningitidis or a Haemophilus influenza, which culture medium is absolutely essential to include in the setup. A. Blood agar. B. McConkey agar. C. Chocolate agar. D. Mannitol salt agar. E. Lowenstein-Jensen medium.
The answer has to be C, chocolate agar.
Why not just blood agar?
Because H. influenzae and Neisseria are fastidious. They require specific growth factors. Factor V, NAD, and factor X, hemin, which are located inside red blood cells. These bacteria are not hemolytic. They cannot break open the red blood cells on their own. Chocolate agar is made by heating blood agar, which lyses the red blood cells and releases these essential factors into the medium, making them available to the bacteria. They would not grow, or would grow very poorly, on standard blood agar A.
Makes perfect sense. Question 4. A 25-year-old male presents with a single, painless, indurated ulcer on his penis. A swab of the serous exudate from the lesion is taken for a Gram stain, but no organisms are seen. Which microscopic technique would be the most appropriate next step to visualize the causative agent directly from the lesion? A. Phase contrast microscopy. B. Dark field microscopy. C. Electron microscopy. D. India ink preparation. E. Giemsa stain.
The correct choice is B, dark field microscopy.
Explain that one.
The clinical presentation is classic for a primary syphilitic chancre caused by the Spirochete treponema pallidum. Spirochetes are incredibly thin, typically less than 0.2 micrometers in diameter, which is below the resolving power of a standard bright field microscope. That's why the Gram stain was negative. Dark field microscopy uses oblique lighting that causes these thin organisms to scatter light, making them appear as bright modal corkscrews against a black background. It's a classic method for direct visualization.
Great. Last question. Question 5. A hospitalized patient has 4 blood culture bottles drawn. After 24 hours, the lab calls to report that one of the 4 bottles, a single aerobic bottle, is growing Staphylococcus epidermidis. The patient is a febrile and clinically stable. What is the most likely clinical interpretation of this result? A. The patient has infective endocarditis. B. This represents a catheter-related bloodstream infection. C. This is likely a contaminant from skin flora during collection. D. The patient is developing septic shock. E. The lab has made an error in identification.
The most likely interpretation is C, contamination with skin flora.
And the logic here is based on probability.
Exactly. Staphylococcus epidermidis is the most common commensal bacterium on human skin. Growth in only a single bottle out of a set of 4 strongly suggests that a small number of bacteria were introduced from the skin during the needle puncture. True bacteremia or sepsis would typically result in a higher bacterial load in the blood, leading to positive results in multiple bottles, often both aerobic and anaerobic. Given that the patient is stable, contamination is by far the most plausible explanation.
Fantastic work. To really consolidate all of this, our recommendation is to open up Murray's Medical Microbiology, Chapter 4. Don't just read it. Look closely at the pictures of the stains and the colony morphologies on the different agars. Visual memory is absolutely everything here.
And then cross-reference the clinical interpretation principles in Jowett's Chapter 47. Remember, a lab result is not a diagnosis. It's a clue that needs to be interpreted.
We'll see you in the next deep dive.